Now comes the part of the specific nucleotide change that can cause different, again, different types of mutations. If you have, for example, here a gene, this is the gene for a certain protein with 4 amino acids, that's a polypeptide, and then a stop codon, UAA. Now, if you have a mutation, usually in the 3rd amino acid of a codon, now why? Why is it in the 3rd amino acid of a codon? What is that 3rd amino acid here of the codon? That's the WABO, or the WABO base, the 3rd amino acid, which is, you get that codon. You have 1, 2, 3 positions. The 3rd, this one here is the one that doesn't bind too strongly to the tRNA anticodon. And then this one is important, that one is important. So usually you have this degenerate code, or this redundancy on the code, that even if a mutation happens here, you're still able to put the right amino acid as if it was the original sequence without the mutation. So that's a form of avoiding serious problems due to DNA mutation. Now, if it happens on the 3rd nucleotide of the codon, it's usually a silent mutation. And what that is, as the name says, is a mutation that doesn't say much. It's a quiet mutation. So it got a mutation, yeah, but it doesn't really say much because you got the same protein that you got without the mutation with the original sequence. So when you have a mutation that doesn't do anything, it's silent. Now, when you have a mutation that is either on the 1st or the 2nd, usually on the 1st base of the codon, usually you have a mutation that will cause some change to the codon and to the recognition by the anticodon. So here, instead of bringing, let's say, the tRNA for proline, you change the 1st base, you're going to bring the tRNA for alanine. Now, you do have a change in the protein, because when you go to your protein expression, as you have here, see, in this case was glycine, you change the 1st base, now you get serine instead of glycine. So this is not a silence mutation. This is something that changed. This is a missense mutation. You can imagine your protein was making a lot of sense before it got mutated, and then you got a mutation and you got, missense, got a missense and then mutation. Still you have a certain size of a protein, which is the same as the original gene, but now it has 1 amino acid that is just out of place. Is there and it shouldn't be there. It should be a different one. That is a missense mutation. Then examples of these cases of missense mutation are sickle cell anemia and also cystic fibrosis. Remember we're studying about the channel, the calcium channel for cystic fibrosis that does not work well because of a mutation. Now you see that in order to have a defective protein or defective channel, there was a missense mutation. And then if you have heterozygous alleles, one with the normal and one with the mutation, usually the person is okay because the normal one will pick up the function. Remember that, cystic fibrosis? Also, Tay-Sachs disease. Now see, these diseases that are caused by missense mutation are usually a lot of problem if you inherit the homozygous form of the alleles. If you have heterozygous, you're usually okay. But in the type of missense mutation, if you have homozygous, then it's a problem. Like in Tay-Sachs disease, remember, homozygous alleles are lethal. The combination is lethal. It usually will kill before age 3. Now, there is another type of mutation, as you look here, in this codon for lysine. Here is AAG. And then if you get a mutation here on the 1st base, and now you change this code to UAG, now this becomes a stop codon, which was that case that we had here. UGA, UAG, got a stop codon. So now, instead of having this protein that was going to be translated, you stop it right here. So you don't have that. You have a truncated protein that usually doesn't work. So you have some type of a loss of function or truncated protein, and that is something that you can imagine doesn't make sense. It's not going to work. So that's a nonsense mutation. If I ask you here, Okay, I'm not talking about a question in the test here. I'm talking about reasoning question. If there is a mutation in a sequence of the gene, and you get a different amino acid than the original one that you were expecting, let me say here, okay, you're expecting amino acid 1, and then you get a mutation here, either on the first or the second place of the codon, and now you get amino acid 2 there. And then you look at the protein, what type of mutation is that? It's not a stop codon. If it's not a stop codon, it's not nonsense, so it has to be a missense. Now, if that happens and you get from one amino acid to the second amino acid, but then you look at the protein and the protein works just as well. So what do you call that? No, it's not silent because you changed the amino acids. Yes, there is a specific name for that mutation, and yes, neutral mutation. Did you look it up just now? Okay. Okay, so here what happens is that you're supposed to have a mutation. It is type of missense mutation, but it's not missense because you didn't miss anything on that protein. So if you get the protein with the same aspects, the same properties as the original one, you just have something that, well, was supposed to mess it up and it didn't. It was a neutral attack on this type of DNA sequence. This is a neutral mutation. It did not have anything to do with the function of the protein, but it's still a different protein, so it is a missense. It is a neutral missense mutation. Now, when you look at the chromosomes, like right at the beginning I said you had 2 types of gene, DNA mutation. You could have a chromosomal mutation or DNA mutation. We just look now at the specifics of what happened on the DNA mutation. So you can have, also something that is very important for DNA mutation at this level is that you can have addition of a base or deletion of a base, which I will also go into detail. Now, if you add a base here, for example, there, what's going to happen to that protein? You have bases, 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 and now you remember every 3 amino acids, you have one codon, one codon, one codon. Now, this is your codon, and if something gets stuck in the middle here, now you're going to shift the frame of this codon to this side here. Now, these are the first 3 codons after that reading frame, continuing the reading frame. So when you add a base in here, you have a frame shift on the codon. Remember, like the dog beat the man, and then the big dog beat the man? You have this shift in the reading frames that will just not make sense for the rest of the protein, but you have also codons that are there and can be read, so you're going to have a mutant type of a protein that's going to be translated. Then if it happens that you add 3 bases here, then you're going to have an extra amino acid, but you're going to put the frame shift back into frame. Right, so you can have additions of bases, or you can have deletions of bases. We're going to go into more details on that too. Now, when you're talking about chromosome mutation, we are talking more about usually either a whole chromosome or a huge chunk of a chromosome that can mutate to a different part. When you say that the cells are aneuploidal, remember when we had the HeLa lab, and I bet nobody found 46. Everything was like either 100 something or 3, 4 chromosomes. So those are aneuploidal. They do not have the right number of chromosomes that you're expecting. Or there's another type of chromosomal arrangement or mutations that is called polyploidal. In this case, the cells will end up with extra sets of chromosomes, not only one chromosome that shows up or another chromosome that shows up. So in our case, instead of having 2 times the 23, like 2 pairs of 23 types of chromosomes, in this case, you can have 3 pairs of 23. That would be a polyploidy, 3N instead of 2N. You have a whole set of chromosomes that get together into that cell. That usually, as you're going to see, does not happen with animals. It happens more with plants. Now here's a representation for chromosomal rearrangement or mutation. So at first you can have a whole piece of the chromosome that can be mutated or added or deleted. In this case was duplicated. We're going to go into more detail, don't panic. So this is a chromosomal rearrangement. Or here, if you look at the chromosomes here, this species has 2N, okay, 2N for the chromosomal number, or 6. If you look at this cell here and you find that most of the chromosomes are 2N, but then one of them is 3. So now you don't have a rearrangement. You gain the chromosome out of somewhere. And does this remind you of any syndrome? Yes, Down syndrome, chromosome 21, trisomy 21. So this is a type of aneuploidy. Okay, trisomy is a type of aneuploidy. Now, if you look at a cell and you find instead of 2N, 3N for all the chromosomes, then you say this is a polyploidy, polyploidal state.
Is that possible?
No, it's just not viable. If it would happen, it wouldn't survive. It would be embryonically lethal. And most of animals would not have this, mainly in plants. Actually, this type of chromosomal arrangement is very useful for development of genetically engineered products, of crops. So you can make like fruits without seeds.